Thank you. I'd like to thank the organisers for having me here to present this data. That is a large collaboration with the British Columbia Centre for Disease Control and also the British Columbia Centre for Excellence in HIV and AIDS. So this is just a small proportion of the work that we've been doing in the Ithaca study, and we'll be looking at phylogenetics of hepatitis C in the VITAS study. So Jason's already presented this slide today, and I think it gives a good idea in terms of the contribution of people that inject drugs to new infections for hepatitis C. But you also get the idea that chronic infections that are largely associated with people that inject drugs or used to inject drugs. So we tend to have a good idea of why people seroconvert or associated with incident infection, and also why people that injected drugs were chronically infected. But the question is, what happens once they're infected? What's the smaller level kind of process? So there's a high prevalence of hepatitis C in people that inject drugs, wherever you look at it, compared to HIV infection. So 60 to 80% versus 20%, or in Australia it's about 1% HIV infected. There's also a higher risk per injection event for acquiring hepatitis C as opposed to HIV. So we really need to understand what's happening on a smaller level rather than just a blanket response in terms of these factors are associated with incident infection. And as you can see here, that work done in Baltimore, where hepatitis C kind of gradually decreased over the time period from the late 80s through to the end of the 2000s, but HIV went from low to almost negligible levels. So there must be something that we're missing out and something else that we can do to help prevent these transmissions in the community. The work that we are doing is in phylogenetics, and we appreciate that not many people understand what phylogenetics is, which means you can kind of work your way around it. But I'll just give you a quick little background to it. Phylogenetics is essentially comparing the viral isolates of different samples of patients in time. And if you wanted an easy version, you could look at animals, and then you can kind of separate them into little clusters. And that's a simple thing to do when you have visual differences. But if you try to look at viruses, it's a lot more difficult. You can't look at two different viruses, even under a microscope, and say that they're different. So you essentially have to go to the genetic level to kind of identify their separation. So here you can see in red transmissions from animal hosts into human hosts for HIV, and that was done purely based on genetic analysis. Work can also be done to look in hepatitis C. This is an example of clusters or transmission clusters in acute hep C in Australia. And it kind of shows patients that are very related to each other. So the only problem with this analysis is that you cannot say, as much as we'd like to say, that one patient transmitted to the next, but you can say that they're all related in some way. The only concern with something like this, with a study such as this, is you don't have 100% coverage of sampling. So you don't know how many patients were in between two patients that you've sampled. So keeping this in mind, that we would like to have investigate and characterise trends of phylogenetic clusters and pairs in the VITAS study in Vancouver. It also looked at factors associated with being in a cluster or a pair in this population. So the VITAS study was undertaken in Vancouver. Was anybody that had injected in the previous month living in Vancouver could be enrolled into the study. The subjects were enrolled through self-referral or street outreach, and was prospective open study from 1996 to 2010. So the majority of participants were enrolled in the first 3 years. But there was a secondary enrolment that happened from 2005 onwards. The follow-up for this study was a baseline and then every 6 monthly questionnaire and sample. The questionnaire was very broad and covered drug use characteristics, and also lending and borrowing of syringes and other equipment, incarceration, housing, and other drug and alcohol treatments. The method that we used was to assess a region of the virus discussed already by Andrea that covers a variable and a conserved part of the genome. This allowed us to get an idea of the patient's not only long-term but recent infections to see if they're related to each other. The analysis used a maximum likelihood with 1,000 bootstrap replicates to give us an idea of the reliability of these clusters and pairs. Clusters and pairs were identified using another method called Phylopart, based on the maximum likelihood tree. And factors associated with being in a cluster or a pair were logistic regression using Stata. So to give you an idea of the patients that we had in our study, there was just over 2,700 patients. The majority, as you expect, were HCV positive, and the remaining were HCV negative. Of the nearly 940 that were negative, 122 seroconverted. So combining the available samples, we had just over 1,300 patients enrolled to give just over 1,000 that were RNA positive, so a clearance rate of about 25%, as you'd expect. And from this, we were able to obtain 657 sequences that covered all genotypes, but the ones that we were interested in, genotype 1A and genotype 3A. So we had a higher proportion of genotype 3A than Andrea presented, but that may just be the effect of our cohort versus the diagnostic samples received. Just a quick background on the characteristics of all the patients enrolled. So about 25% were female, which should be expected. We also had an average age of 36 and 15 years of injecting. So these people were long-term injectors in the population, especially enrolled from 1996 onwards. There was about 40% were 3A infected, and HIV infection was about a quarter. In terms of drug use patterns, the high amount of cocaine injecting and heroin injecting, and a reasonable amount of speedball injecting, but not much methamphetamine usage. So not many people would have experienced what a phylogenetic tree is. This is essentially a demonstration on the relationship between two viruses. So you may take, as we've got here, about 300 of the genotype 1A, and we've put in reference sequences. So what is in blue are our reference sequences, and then anything in red is a virus participant that was not in a cluster. Anything that is in yellow is a virus participant in a pair, and anything in green is a virus participant in a cluster, which is 3 people or more. So you can see that even though participants may not, in red, may not actually cluster, be defined as a cluster, they're still very much co-located with people that are in a pair or a cluster, suggesting there's a local expansion of this virus. So potentially the virus was introduced at some stage and expanding the population. Of particular interest in this 1A is a small cluster of 7 that I'll talk about in one moment. Having a look at genotype 3A, we have many less reference sequences. So we had 700 reference sequences for genotype 1A, but in this we only have 60 reference sequences to our 190 virus participants. So that may affect our results, but we're also hoping to add in more reference sequences. And you can see here that there's a large expansion in the bottom left-hand corner. So our particularly interesting, go to clustering. So that about 60% of participants are not in a cluster, which you'd expect. And then there's a high proportion of people in pairs, and then kind of dropping off in tails. And for the genotype 3A, we had the large cluster of 23 participants. In the genotype 1A that we discussed, of particular interest, you can see here that the circles are females, filled in HIV positive and not filled in negative. And then we've got colour coding as well for ages. So of particular interest with this cluster is the high proportion of females. 5 of the 7 are female, where you'll remember that only 25% in the general population were female. And also a high proportion of HIV in this. So this particular cluster that was collected, the patients were sampled in the majority in 1996 or 1997, and there were 2 participants that were acute, so one in 1998 and one in 1997. So we only know the actual date of infection for 2 participants, but you may try and assume that the other participants were also recently infected because of the very short genetic distance. So combining genotype 1A and 3A, we've also looked at those in a pair and a cluster and combined those in a pair and a cluster to be equivalent and compared them to not being in a cluster. And what's shown here are the factors that we found to be significant. So when we did a univariate analysis, the only factors that were significant were younger age and genotype 3A infection. However, when you combine it into a multivariate analysis, you find that HIV infection and acute hep C infection and syringe borrowing in the last 6 months are significant. So these factors are potentially factors that we can use in the next step to identify participants that are likely to be transmitting. So in conclusion, we've found that there's a high proportion of participants that are clustering, so 35 to 45%, and that some larger clusters display unique characteristics that may be useful for us to identify. And in general, being in a pair or a cluster is associated with being younger, being infected with genotype 3A versus 1A. Recent syringe borrowing, which may go on with acute infection. So what we'd suggest in the future is that knowing this information may be useful for public health interventions. So as Andrew has mentioned, the real-time testing and phylogenetic analysis to identify those at risk of transmitting. So further strategies to be evaluated to identify the high-risk networks that are continuing the spread of the virus in the community. And acknowledgements to the virus study participants and the coordinators, and also the people at the Kirby Institute, the CDC and the CFE, and the funding, the Canadian Institute of Health Research.
Thank you very much. Are there any questions? No. Wait. So you're trying to do this for public health, and you're doing this work anonymously, and you showed the correlations that you'd expect. So how would you use this? How would you help public health use this information?
So one thing that we've been considering is the application, as Andrew had suggested, in the real-time testing and looking at phylogenetic, so perhaps a nurse-led model for intervention, so people that have been diagnosed with recent infection and looking at the age category and whether they're HIV infected as well. So obviously syringe sharing is not reported in a diagnostic test, but all of the others you can get from basic demographics.
Any other questions? Thank you.
Thanks.
So